Just like a city with its variety of structures, each with a defined function, the eukaryotic cell is a complex collection of different organelles, a microscopic metropolis in which each organelle is a separate entity carrying out one or other cellular function: information coding and transfer, energy production, synthesis or breakdown of macromolecules.
But hang on, that can't be the whole story. The cell has to function as a whole. So how separate, how autonomous can organelles really be? Surely they must interact to some extent. There must be some overall integration. Well, to investigate this, we're going to concentrate on just one organelle, the chloroplast. That's the organelle characteristic of plant cells.
Perhaps the major evolutionary triumph of green plants is their ability to harness the energy of sunlight during photosynthesis. It's in the chloroplast that the cellular machinery for photosynthesis is located. The organelle, one of a group called plastids, is a complex one. It has an envelope with two membranes, an outer and an inner one. These surround a soluble fraction, or stroma, that contains the enzymes for carbon dioxide fixation. Within the stroma is a system of membrane sacs called thylakoids, which contain chlorophyll. These are organized into stacks called grana, where light is trapped and the energy used to make ATP.
Besides these reactions of photosynthesis, the chloroplast also contains other metabolic pathways. For instance, pathways for the synthesis of amino acids and fatty acids, and others for the synthesis of photosynthetic pigments, such as carotenoids and chlorophylls. It almost seems as if the chloroplast is as complex as a complete cell. And indeed, it also contains DNA, and the enzymes for transcribing DNA to give messenger RNA, and ribosomes and other factors for translating messenger RNA to produce proteins. In other words, the chloroplast contains a complete protein synthesizing system, a complete genetic system.
So what's the precise role of this genetic system? A question that's fascinated John Ellis and his colleagues at Warwick University for many years. Peas are their plants of choice because they're easy to grow and are a convenient source of chloroplasts. But building up a detailed picture of any organelle often calls for some judicious breaking down of the cell, for cell fractionation.
It's clear from the microscopic studies that have been carried out on cells over many decades that cells are very complicated structures. They have lots of internal bits and pieces, and these have been well described by microscopists. But sadly, you can't tell very much from a picture about what these structures actually do. So the purpose of cell fractionation is to actually break the cell open and isolate all the bits and pieces, find out what they're made of, and find out what they do. In many ways, the art of biochemistry is the art of controlled violence, because what we're trying to do is to break the cell open to release the various bits and pieces, but not be so violent that you destroy the properties of those pieces. So essentially we aim to try and get out chloroplasts which are intact, because that approximates more to the normal in vivo situation.
A crude chloroplast suspension can be obtained from the leaf homogenate by centrifugation. With practice, it's possible to obtain suspensions for experiments in less than 5 minutes, a crucial factor when trying to isolate metabolically active organelles. After centrifugation, the top layer contains the soluble components of the cell which can be discarded. The remaining dark green residue is a mixture of intact and broken chloroplasts. So much for isolating the organelle. What about its structure? One approach is to examine its component proteins. A useful method for this is known as SDS gel electrophoresis. Samples of chloroplasts are loaded onto a gel containing the detergent sodium didecyl sulfate, or SDS. This separates the proteins in the samples into their component polypeptide chains. Expose the samples to an electric current, and the protein molecules migrate down the gel at a speed determined by their size. A variety of chloroplast preparations can be studied in this way: the thylakoids, the stroma, or the whole organelles. After staining the gel with a blue dye, the proteins show up as individual bands along each sample track. In an SDS gel, the proteins are separated according to molecular mass, with those of high molecular mass migrating more slowly than those of low molecular mass. Remember, there are 3 sets of tracks, one for whole chloroplasts, the stroma, and the thylakoids. There are many proteins distributed between the chloroplast fractions, but the dominant bands turn out to be components of a single protein in the stroma, rubisco. One, with a molecular mass of about 55,000, is called the large subunit. The other, with a molecular mass of 14,000, is called the small subunit. The rubisco molecule in fact contains 8 copies of each type of subunit. Rubisco stands for ribulose bisphosphate carboxylase oxygenase. It catalyzes an important reaction in the stroma of chloroplasts, the addition of carbon dioxide to the 5-carbon sugar, ribulose 1,5-bisphosphate, to form 2 molecules of a 3-carbon sugar, 3-phosphoglycerate. This is the first step in the photosynthetic cycle that fixes carbon dioxide into carbohydrates. Rubisco is the most abundant protein in chloroplasts, making up about 50% of their total protein.
Now, abundance works in your favour if you're a biochemist. It means that the protein is easy to detect in extracts. It means it's very easy to characterise and purify. But more importantly, it means that the genetic system of the plant must devote a large part of its effort to making this particular protein. So the gene for this protein is active, and the messenger RNA for this protein is active.
Okay, so rubisco is the major chloroplast protein. Now remember that chloroplasts contain DNA, ribosomes, and all the various enzymes needed to synthesize RNA and proteins. So it's pretty obvious that the major chloroplast protein, rubisco, must be synthesized in the chloroplast.
The best way to study this is to look at protein synthesis within the chloroplasts themselves. First, any intact chloroplasts must be separated from those with broken envelope membranes so that the ribosomes in the stroma can be retained. This can be done by layering the crude chloroplast suspension onto a gradient which will separate the different components according to their buoyant densities. After centrifugation, the chloroplast mixture becomes resolved into a series of bands. The lowest layer contains broken chloroplasts, while the central layer contains the intact organelles. Do these actually synthesize their own proteins? We can follow this by adding a labeled amino acid, such as methionine, to see if it gets incorporated into newly synthesized protein. In an illuminated water bath, The intact chloroplasts use light energy to generate their own ATP, which then drives protein synthesis. Remember the total array of chloroplast proteins revealed by the SDS gels. Which of these contain radioactive label and so were actually synthesized during the experiment? To find out, some radiation-sensitive film can be placed over the dried gel. Any radioactivity in the sample will affect silver ions in the film emulsion. Developing the film gives an autoradiograph whose pattern of darkened silver grains will reveal the distribution of label within the sample. Remember the SDS gels which showed the proteins present in whole chloroplasts, the stroma and the thylakoids. Here are the large and small subunits of Rubisco. Now here's the autoradiograph showing which proteins were actually being synthesized by the chloroplasts. In the stroma, the main product is the large subunit of Rubisco, but there's no trace of the small subunit being synthesized by the chloroplasts. So where is it actually made? One way to find out is to examine the effect of antibiotics on protein synthesis in intact shoots. One is placed into a control tube containing labelled amino acids. A second is placed in a tube which also contains chloramphenicol. This inhibits protein synthesis on chloroplast ribosomes, but not on ribosomes in the cytosol. A third sample is placed in cyclohexamide, which has the reverse effect. It inhibits protein synthesis on cytosolic, but not chloroplast ribosomes.
The advantages of using inhibitors for studying what proteins are made by chloroplasts is that you can address this question without disrupting the cell, and providing you have a suitable way of monitoring the synthesis of the chloroplast proteins, you can then use these inhibitors to determine which part of the cell is actually making a particular protein.
So what are the effects of the antibiotics on the young plant cells? Remember that chloramphenicol inhibits protein synthesis on chloroplast ribosomes. We can use it to follow the synthesis of the large and small subunits of Rubisco. Results show that the synthesis of the large subunit is inhibited, but not the small subunit. The situation is different with cyclohexamide, which inhibits protein synthesis on cytosolic ribosomes. This time, synthesis of the small subunit is inhibited, but not the large subunit. So these studies suggest that the large subunit is synthesized in the chloroplast, while the small subunit is made in the cytosol.
So as it turns out, Rubisco isn't entirely made in the chloroplast. Large subunit is made there, but small subunit isn't. In fact, small subunit is synthesized in the cytosol, on cytosolic ribosomes. Now that must mean to get entire mature Rubisco in the chloroplast, the small subunit must be transferred from the cytosol into the chloroplast. But how?
The small subunit is made in the cytosol in the form of a precursor. That is, it's slightly larger in size than the mature small subunit that you isolate from the mature Rubisco. The reason it is larger in size is because it has an extra set of amino acids at one end, and this set of amino acids acts essentially as an address. It tells the precursor to go to the chloroplast, and when it's at the chloroplast, it allows the chloroplast to take it up. Once it's inside, the extra piece is removed, thus generating the mature small subunit inside the chloroplast, where it then assembles with the large subunit to make the complete Rubisco.
But assembly of the subunits within the chloroplast to give the mature enzyme isn't straightforward.
We observed in our early work on the synthesis of the large subunit of Rubisco by isolated chloroplasts that the large subunit, when it's made, does not enter into combination with the small subunit immediately. Instead, it combines with another protein. And the purpose of this protein, we think, is to ensure that the large subunit interacts in a correct fashion with the small subunit. If this protein isn't present, then what happens is that the large subunit interacts with itself incorrectly to produce a useless product. And so for this reason, we've termed this other protein a chaperone. It's rather like the human chaperone. The function of the human chaperone is to make sure that a pair of people interact together in a proper fashion. But once that job is done, then of course the chaperone goes away. The chaperone isn't present on the wedding night, for example. And therefore this seems an appropriate name to describe the function of this type of protein.
So we've seen that in a leaf cell, the components of rubisco are coded for at two distinct sites. The DNA of the chloroplast specifies the information for the large subunit, which is synthesized on chloroplast ribosomes. However, the DNA coding for the small subunit is located in the nucleus, and this subunit is synthesized on cytosolic ribosomes. The small subunit contains an address label that allows it to be transported into the chloroplast. Once inside, the address label is removed. A special protein, called a chaperone, is also synthesized in the cytosol. It too contains an address label that enables it to enter the chloroplast. By binding to the large subunit, the chaperone assists in the assembly of the small and large subunits into the whole enzyme.
So the chloroplast does have a degree of autonomy. But despite its own genetic system, it can't even make all its own proteins. And we could have told a similar story for the mitochondrion. That's also got a complete genetic system, but like the chloroplast, it too can't make all its own proteins. So why are things organised this way? Why this partial autonomy? Well, we could ask a similar question about some cities. In many ways, this is a typical village. A church, a green, a high street with a few shops, and of course a pub or two. A little self-contained community out in the countryside. Well, in fact, it's not in the countryside. It is a village, Highgate Village, but it's right in the heart of London. If you didn't know London, you might think that it was deliberately organised this way. A cluster of semi-autonomous villages, each with their own shops, serving the needs of the local people. But the main reason is actually historical. About 200 years ago, Highgate was a village, several miles outside London. But the city gradually swallowed it up, so that now it's part of the busy metropolis. Now back to cells and organelles. When we see the apparent semi-autonomy of chloroplasts and mitochondria, should we be looking for some functional reason for this way of organisation? Perhaps some subtle economy to the cell as a whole? Or, as for Highgate, should we look more towards history, towards an evolutionary explanation?
From our present knowledge, it doesn't make a lot of economic sense to have genes outside the nucleus. It would seem a lot simpler to have all the genes inside the nucleus. And so, as with many things in biology, we have to turn to the history of the system. And the current belief is that this situation has arisen because the eukaryotic cell itself is really a mixture of several types of cells, including prokaryotic cells, bacterial cells. In other words, if you go back in evolution far enough, you would find a situation where there were eukaryotic cells, but they didn't have any mitochondria or plastids. These arose subsequently by the development from ingested symbiotic bacteria.
According to the hypothesis, free-living photosynthetic bacteria, which probably resembled our modern-day cyanobacteria, were actually taken up by primitive nucleated cells over 1 billion years ago. The bacteria retained their ability to photosynthesize and to replicate throughout successive generations of the host cell. But eventually they lost their capacity for independent survival and became an integral part of the host. This probably involved a transfer of some genes from the ingested cell to the host nucleus. This process of endosymbiosis could represent the first step in the evolution of organelles such as the chloroplast. Similar arguments are used to explain the origin of mitochondria. So that's the story. But what evidence is there that chloroplasts might have had a bacterial origin? Let's start with the molecular evidence. The circular DNA of the chloroplast is very similar in sequence and arrangement to bacterial DNA, and quite different from that in the nucleus. Chloroplast ribosomes are also closely related to bacterial ribosomes. They're even sensitive to antibiotics like chloramphenicol, which are well-known inhibitors of protein synthesis in bacteria. But we don't even have to probe as deeply as that. This flatworm is green because it harbours photosynthetic algae in its tissues. The intimate association between two organisms, symbiosis, is widespread in nature. In fact, many single-celled organisms are found as symbionts in a variety of invertebrates. What's more, endosymbiosis, one cell living inside another, also occurs. But we're interested in what might be the next evolutionary step from this. Here's the unicellular organism Cyanophora paradoxa. It contains structures called cyanelles, whose cell walls and photosynthetic membranes are remarkably like those of cyanobacteria. But unlike free-living cyanobacteria, these cyanelles don't have enough DNA to code for all their proteins. In fact, they only contain about as much DNA as a chloroplast. And yet, unlike modern-day chloroplasts, the cyanelles do have genes coding for both the large and small subunits of rubisco. So it's tantalizing to speculate that these structures might simulate an intermediate stage in the evolution of chloroplasts.
Our study of the chloroplast has covered a lot of ground. At the practical level, we've seen the importance of both cell fractionation and the use of inhibitors in dissecting a cell. In other words, finding out where things happen. And at the biological level too, there's been a very important message. Often, when we're looking for explanation in biology, we have to resort to history, to the evolutionary history of the system.